Professor Walker, thank you so much for that inspiring and interesting talk. We have so many comments from the audience members, how you've really inspired them, how it has been very interesting. Also, going back to your roots to help out your community, that's definitely something we all should keep in mind as scientists. At the end of the day, it's all about betterment of life on this planet. A lot of questions coming in. I'll try my best to combine a few of them together so we can answer as many as possible. Getting started with that first tale of natural product about asadirac, complexity of this compound. How did you extract it from neem seeds?
Yeah, it's a methanolic extract. And then we have a subsequent HPLC approach, which is a macroscopic amount. But what was used in the field trial was actually, it's a powdered extract, which is methanolic. And then what we did was we used a variety of different carriers in order to get it to go properly onto the pellets. What we needed to do was we needed to pay attention to having a food grade solvent that we could use that then also wouldn't make the fish think it would smell not so good. So I hope that helps to answer.
And also one more question on that part about the pesticides that were being used on the lice and parasites. So how did we ensure those compounds are not harming the fish,
right,
the eventual consumers of the fish?
Yeah. So what we did was we analyzed the fish. We had controls. If you see, we looked at fish of different that were being fed different things. Some of them were just being fed regular food. Some were being fed food that was treated with the solvent, but not the neem extract. Some of them, and then we had a statistically large enough number so that we could do the analysis. And everything that we did was then actually analyzed by Health Canada, which is a group inside. And we got feedback from them on this process. It was not an official feedback procedure, but it was nonetheless, that's what happened. And then we were required to save the fish. We took the fish and we did HPLC on tissue residues. In fact, even now I have many freezers full of fish in my laboratory, which is not very popular with my boss, by the way. But nonetheless, we then analyzed for residues of azadirachtin and the other components, and showed that they disappeared basically by hydrolysis within several days. So we don't have to wait a long time after the material is withdrawn from the feed, and then so within a minimum, I forget the exact period of time, but I think we predicted that within 3 weeks after the treatment there would not be any issue whatsoever. And as many people on the Zoom probably realize, neem is not poisonous, and it's actually widely used as a pharmaceutical for many applications in traditional medicine. And I've drunk neem solutions myself, and for purposes. So I think it's not a big. I personally don't think it's a big challenge.
Well, if you could forget about the taste of it, right, Professor Walker?
Yeah, it has an interesting taste, but you should drink. If you think neem oil tastes strange, I invite you to drink karanja oil.
Okay. All right.
That's less tasty. But it has a very similar effect. And if you're interested in this space, I encourage people to actually see what opportunities there are from karanja oil. There are some very interesting opportunities.
Definitely. Thank you, Professor Walker. Some interesting questions have come in about chitosan, although they might not be directly related to your research, about what are some of the other sources of chitosan, and is there a recommended daily level that adults can kind of tolerate without any toxic effects? And does chitosan lower creatinine?
Yeah, those are all very good questions. And I don't know how much chitosan lowers creatinine. I would point out that the amount that we're delivering here is very, very small. It's at the level of milligrams per kilogram, so it's a very small, I mean, per body weight, it's very, very low quantity. My mother consumes chitosan at a much, much larger amount than that, that's for sure. But where do we get it? There are different sources, as I mentioned. There are fungal, and there are also like crustacean sources. We've used both. The paper there, I think, was oriented from a fungal source.
Thank you, Professor Walker. There has been a lot of questions on the microgels, so I'll combine a few of them with common themes. One question is, why was these made of alginate? Why was alginate chosen as the medium? And can you suggest some other polymers that could also serve the purpose?
So alginate, yeah, right. So in the gel, you would like to have a gel which is a hydrogel, so it has mostly water content. You would like to have one that you can control the stiffness, and you would like to have one that has a good capacity to allow the cells to develop in a healthy manner. I find that the alginate is very good in this context because you can change the calcium concentration, and by changing the calcium concentration, you can change the degree of cross-linking. So you can mimic different environments which will have different stiffnesses or different elasticities that way. The reason that I liked, so the Gelma is it's known that alginate by itself has limitations because it doesn't, for example, have such strong interactions with many cell surface receptors. It has some, but different types than, for example, Gelma. One reason that I liked including the Gelma in combination with the alginate, and I think it's really nice to think about combination gels. The reason that I think about it is because you now have two interpenetrating networks, which is a very interesting area of research. Interpenetrating networks are a relatively new area of polymer science, and they allow you to control the stiffness of the material in a very different way. And I really encourage people, to think about how to combine, for example, two different polymer types to make a gel. If somebody wants to have a list, please feel free to send me an email, and I'll try to give you a few. Okay. My email is very easy, gilbert.walker@utoronto.ca. Okay, very easy.
Thank you, Professor Walker. Another question that came in was about what are the challenges that you have faced in using these alginate-based microgels, particularly in terms of their fabrication process or efficiency?
Right. So it was important when we were working with these alginates to get the flow in the system correct to begin with. As you saw, that microfluidic device basically creates a small droplet, and we bring those, or two small droplets that are then brought together, and they cross-link. One thing that we've been trying to understand in greater detail, and I have to confess that we don't understand it as well as I would like to yet, is if we have these two gels, here, let my hand be one gel, and here's another gel, we don't really have a great understanding of the interpenetration between the two, this boundary between the two. And that's obviously going to play a role as we talk about, the cell moving from one side to the other. We don't currently have a great way of understanding that in detail. Second thing is, it is possible if you do the flow through the microfluidic device too fast, so to sort of tear the gel. So you have to go slowly enough so that you don't tear the gel. Of course, you can overcome that by making a much stiffer gel, which is what we initially did. But then a much stiffer gel is not a good host for the cells. So those are a couple of the challenges that we faced. again, if you are interested in this space, I invite you to drop an email and be very happy to give some comments, additional comments.
Sure. Yet another question. We have a lot of safety conscious people today in the audience. Could you please comment on the safety aspect of these type of microgels in terms of compatibility to?
So these gels are being used not internally to people's bodies at the moment, I don't believe, although there are examples in clinical trials. They are currently being used in topical applications. So basically, for example, people who have an issue with their skin, you can actually make these gels and then you can do a transdermal delivery through that. As I said, there can be an undesired immune response if the alginate is not adequately purified. That's really an issue. And we found our immune data to be kind of strange at the beginning because we weren't adequately purifying our alginate. And so we wasted probably a month figuring out what was going on there. And so that's another issue. You know, implants, people have been working on implants for, let's just take the example of artificial pancreas, right? Since the 1980s, people have been making these implants, and there are a lot of long-term consequences which are not easily managed. The long-term consequences, we talked about the immunological ones, but there are other ones that happen just because the implant sort of develops a coating of other kinds of material from the body, proteins and so forth, which change the performance of that. So we have stories that are quite terrible as well, right? We have stories of polydimethylsiloxane implants, which turned out to be very bad for people. So this is really an issue that has to be tested quite carefully. We don't propose at the moment to put these in. For us, this is a testing platform.
Thank you, Professor Walker. We have an audience member who is actually reminiscing quite fondly their PhD time with natural projects and chemical ecology. And they have a question for you. What is your outlook and vision for the field of marine natural products research?
I think there's huge opportunity, huge opportunity. I think that in terms of drug discovery, there are enormous numbers of compounds that have not been explored. in terms of the environment, the marine environment, the environment itself is not something. I mean, unfortunately, there is global warming going on, and that is creating a hotter environment that is, in many cases, leading to the death of species in the oceans. But I find the marine the frontier of marine chemistry one of the most exciting, because it's the region of space which is very much unexplored on our planet. And so I think that's really important. And I think that I really love working on it. One thing I would like to comment on, and probably this questioner realizes, is that phytochemical analysis or marine phytochemical analysis is complicated because you have many isomers, you have many different minor and components inside there. And so you have to, really be careful about developing analytical techniques to separate this. So the risk, I think, is when people publish papers and they don't really specify what they did, because then it's very hard to reproduce. There was a question somewhere along the way that we had at the beginning about how do we extract the compounds. And that's really important, right? Because unless you do the combination, a straightforward, the paper, by the way, is published in Aquaculture in November 2022. I can send a copy to anybody who wants it if you can't get it yourself. I think it's in November of 2022. First author, Chris Kim. In any event, we went to some pains to describe the composition and exactly how we did the extraction and stuff like that. I believe that's really important. One reason is because there are a lot of papers that don't do that carefully, and then it's very hard to reproduce, very, very hard. And so I love the topic, and I think that many of the purification techniques can be informed by large-scale purification techniques, but in the end, you still need to do analytical chemistry in detail, I think. Yeah.
Thank you, Professor Walker, for that insight. So, in the interest of time, I think we will not be able to take more questions. But thank you so much for your time, for your patience, and also for sharing so many unique research questions for the young audience. We would request you to kindly provide your concluding remarks and any advice to our audience members today.
My concluding remarks are, I'm so honored to have been here. Honestly, very honored to have been here. And I hope that what you took away from this in part is how in my life I really maintain a very strong connection to my roots. And I feel, even though I have some projects that really, when I talk to my mother who is 93, she talked to me the other day and she says, "Are you studying electrons?" And I said, "Yes." And she said, "Weren't you studying electrons 40 years ago?" And I said, "Yes, yes, yes." And she said, "When will you change and study something new?" And so sometimes family doesn't get it, right? Okay. But nonetheless, it's valuable to me that by telling her these stories, and by the way, when I did this field trial with the cages in the Bay of Fundy, I would comment that my mother, this is a very small community I come from, 200 people in the village, okay? My mother said, "Don't make any mistakes." That's all she told me. And she kind of frowned at me. And that's because in this small community, it's a risk, right? If you do something and there's an economy of the local economy that depends on things being not a risk to the environment, then it's actually important. So I really, listen to your mothers, okay? Your mothers give you good advice. They say be careful, and then they push you to try new things. So I'm still trying to listen to my mother, and I hope you will too. Thank you very
so
much. I appreciate it so much.
Definitely. Over to you, Kunal.
Oh, thank you so much, Asha. And so thank you so much, Professor Walker, as well. So this brings us to the end of today's broadcast. We hope that you have enjoyed this broadcast. We invite you to view the edited recordings from the past events in the ACS Science Talks library. At the end of the broadcast, you will receive a brief survey. Kindly share your feedback with us so that we can continue to improve and serve you better. On behalf of all of us at ACS, thank you for joining us today. We hope to see you again soon for more ACS events. Until then, stay safe and healthy.